How are the two of you doing today? Good?
Well,
Good. Yeah, why don't you please, tell us, tell the audience your full name and maybe just a quick version of what you're doing in the park. Do you want to go ahead and start, Ricardo?
Yes, hello everyone. I am Ricardo Guta. I work in Gorongosa National Park in Wilson Laboratory as a research technician, and I work as an entomological technician. I used to document the biodiversity of insects.
And how about you, Norina?
Hello everyone. My name is Norina Vincente. I am from Tete, which is located in the center of Mozambique. And I have a bachelor degree in ecotourism and wildlife management. And here in Gorongosa National Park, I'm working as a zoological technician. So I help other scientists in the field, in a lab, but also I work especially with insects. So I collect data, I prepare them, I identify, and I also put them in our database.
So again, you're both from outside of the park, and now you've become part of this community. Can you tell me a little bit about what that means to you and how the park works with folks who live in and around the park?
So, because conservation is not about just protecting an animal. So we need also to see the other parts that are also involved. So we cannot just protect without including humans. So around the park, we have more than 200,000 people. So we need to engage these people to give them better understanding why it is important to have a particular species in this entire ecosystem. So one of the programs is a Girls Club. So we know Mozambique is a poor country, and we know in the community when a female born, sometimes she already have a husband because the father doesn't give her an opportunity to choose. She just born, and then the father just will let her to grow a little bit, and then he just look for a husband for her, because sometimes first people have a lot of child. So when they have a lot of child, they're expecting to one of those, if they work, or for example, if we have a lot of females in the families, meaning that the husband will give me more cows, the husband will give me more money, the husband will build a house for me. So women in our community doesn't have that voice, and we know that women's caregiving. So if we educate women, it's meaning that we are giving opportunity to the next generation be different than what we have now. So the Girls Club, it's a really a huge program that helps girls from around here in the park. So we already have more than 2,000 girls in this program. So, and it's covering more than 50 schools. What I would like to say is conservation is educating people, because only work together we can maintain this entire ecosystem.
Right. So what's happening in Gorongosa is important for all of Mozambique, all of Africa, but actually for the whole world. We're all in this together. It's really good. Yeah.
Exactly. And we hope as a Gorongosa National Park, we hope that what we are doing here can be transmitted to other parts of Mozambique that is in danger. We have different national parks and reserves that is in danger because people just are cutting the trees and other things. And we hope that in the future people can just use use what we are doing in the park as an example of.
Is that the two of you working in the laboratory?
Yes.
Yes.
Yeah, okay, great. So I think that's an important thing. You know, sort of a great aspect of the park is that you have good wet lab facilities placed in the context of this gigantic park. Do you want to tell me a little bit about what are you doing here?
Okay. As a technician in Gorongosa National Park, we are trying to understand what we have in terms of biodiversity. To do that, normally we just go to the field and collecting data, and we use different tools for identification. First of all, we use a taxonomy by using morphology and other characters for identification. But because we have a molecular laboratory here, we also do the extraction and using this data also to confirmation of the species. What you are seeing here is us getting training of molecular, how can we do molecular analysis.
So you collect, if you collect a specimen in the field, you take a sample or you bring the specimen back to the lab, and now you're isolating DNA so that you can do other studies and have a genetic identification of the species. Is that right?
Yes.
Yeah. Okay, that's great. And here's Norina. You appear to be very good at pipetting. You like to pipette. But is this you here in the field?
Yes.
Yeah. So what are you doing here, Norina?
So this was one of the days that we went to the field in a coffee plantation. So we were looking for the vegetation, but also see the coffee. So exactly what I'm seeing here is a species of Pheidole megacephala. So what I was doing here is just collecting to see, and also doing observation to see what are the behavior of the Pheidole megacephala on the species of coffee. Coffee and also other plantations suffer with some pests. So what we are trying to do now is using a biological control to avoid that. So one of the species that we are thinking that is already proved in all world, in terms of Australia, Asia, even here in Africa, is Ecophila longinoda. So we are thinking to use Ecophila longinoda to help the coffee project, because sometimes farmers use chemical, and we know that chemicals are not selective. So rather than kill billions of biodiversity on the soil, it's better to use organic animal, such as an ant, to prevent that.
How can you? Is there a common name for that species that you're planning to use as a? What's the common name?
Fever ant. Yes, weaver ant. Sorry, weaver ant.
Ah, and then
Exactly.
here you're collecting Pheidole. Now, if I recall, that genus is important. That's a genus that Dr. Wilson is particularly interested in. Is that right?
Exactly.
Well, while we're talking about ants, maybe you could just tell me, how did you come to be so interested in ants, and what aspect of ant biology is most interesting to you?
So everything started in when I was in university. So when it was in 2015. So I was seeing videos from two girls from Gorongosa that were studying lions. So, and I was admire myself how two girls can be in the middle of the bush studying such dangerous animal. Then in the next year, I was seeing videos from Professor Wilson talking about ants, and also the biodiversity survey that he did here. then I have this huge opportunity to come here because it was my dream to have just one opportunity to come here and have an internship. So that happened in 2016. So I came here, spent 5 months. So I was working with science department, and also I had opportunity to participate on 2 courses. So one was bioinformatics, which I put in practice what I learned in university. But the most important thing that I learned that I never forget is creating a database of the species that you collect and also how to manage that. But then everything really started when I went to biodiversity survey. So each person had to choose one group to study. And then I was seeing my colleagues choosing other groups such as mammals, amphibians, and I chose study insects. But also I was wondering, people were choosing those that is small, easy to see. And how about the tiny one? Is there any importance, or people are noticing that?
Right.
So then I chose to study ants. In the beginning, it was really hard. Firstly, because English is not my primary language, and that was also my first time to see a microscope, to work with a microscope. So you can imagine that was really hard. But then I get to that, and then I move forward.
So here we have Ricardo working in the field.
Yeah.
Yeah. What are you doing, Ricardo?
I'm trying to collect insects. What you are seeing there is me looking for insects. As you know, we can find insects in the ground, in the grass, and in different ecosystems. And I'm trying to look for grasshopper.
And so my understanding is your particular research interest and focus is in fact on grasshoppers.
Yes, exactly. I'm interested on grasshopper because, as you know, the grasshopper is the main component of the ecosystem, because they help to regulate the vegetation, like grass and other things, and they also is they also are food for other groups of animals like mammals, birds, and some other pest insects. So what I would like to understand in my research is to understand how many how many species of insect that we have in the park, and in further I would like to understand also how many species that we have in Mozambique. And after that, that can help me to answer some ecological question. And also because grasshoppers, for example, in the park we have a buffer zone, and for surviving they like practice agriculture, and grasshoppers used to be pests in agriculture. And for controlling grasshopper, people use a chemical and other things. But we know that the nature can control the pest by yourself. That's why in my research I would like also to include the parasites that associated with the grasshopper, like for example some flies and nematodes, and also some mites. And I would like also to do the documentation like acoustic grasshopper, how many species of acoustic grasshopper that we have in the park. And in this picture, I was working with the top church, looking for termites. As you know, termites is they invented the agriculture, and they also help to maintain the ecosystem by by showing the piece of grass and other organic materials and incorporating the soil, make that fertilize it, that can allow the trees and other kind of vegetation grows, and later the animal use that for feeding, for feeding.
Yeah.
And
Yeah, the termites are like ecosystem engineers. I've seen what they do to the landscape. They literally create, you know, an undulating landscape. Some of the mounds are so big. They're a huge impact. Species, do you have some estimate of how many species there are in the park?
we have about 150 species of grasshopper in the park.
Have you discovered any new ones yourself?
We just discovered 9 species for science last year with Dr. Piotr Naskrek, but that was a candidate that is also in the same group of grasshopper.
Oh, close relative.
Yeah, close relative.
That's so good. Yeah.
Yeah.
How about you?
But
How about when it comes to ants? Have you discovered any new ants?
Well, some that I knew for the regions, but I have some. I think it was in 2018. I was working with one mimicologist from Poznan. So we find one queen from Campanotus that is undescribed. So now we are writing the article.
Mm. That's it. So both documenting, you know, what lives where in the park, and at the same time, when you go out in the park, you never know when you might discover something new that's never been described before. That's really exciting. I don't know about being able to audio record, you know, ants. Maybe there's something interesting there, but I know many insects are known. They're bigger. They have distinctive songs. So, Ricardo, maybe tell us a little bit about acoustic monitoring.
Studying acoustic in animals, it's a special grasshopper is really interesting, because sometimes in the park we have a different ecosystem, different habitats, and some of them it's very close and you cannot like go in the bush, or it's a little bit difficult. But for insects or other groups of animals that can sing, it's a really nice tools to use for identification purpose, because you can just record, and by using a books or other online database that have this information, you can just use that song for the identification. And as I mentioned before, we use for identification, we can use different tools. First of all, it's based on taxonomy, that you can look for morphology, and we can use also molecular data. And this is another tool that we can use, because as we know, each species they have a different calls, and it's really important for identification purpose.
So if I'm understanding how you do these experiments, it seems there'd be several advantages, right? You can set up a microphone like this to monitor a certain area. So that means you can detect things that would be hard to see, or perhaps doubly hard to see because they're more active at night, those sorts of things. So like when you look at one of these, when you look at the data from an audio recording, how many species can you pick out? How can you recognize the different species?
You can recognize different species. First of all, as you know, different groups, they use different character for producing sounds. For example, the grasshoppers, they produce sound by using the hind femur and rubber between the tegment or the wings.
Yeah.
And by doing that, they produce a different kind of the sounds. And if you look like katydid and cricket, they produce sound by rubbing two wings like this.
So grasshoppers like
Yeah,
play the side of their body, and
yeah,
katydids rub their limbs together. Huh.
the wings, yeah, the wings together.
So you can tell sort
And
of general group, and then yeah, very very interesting. And can
yeah,
you even identify? But I assume there you can. In some cases, you can identify a specific species from the sounds.
exactly. Yeah, what we are creating here. First of all, we expect to record every single species that produce sound here in the park. And by doing that, we will also identify the species and put together the image of the species, the sound that they make, and for in the future, because our main, as you know, we cannot preserve, we cannot keep this park well preserved if we don't know what we have in terms of the species.
Right.
So if we record all the species that we have, even someone can come here and just go to the field. Like, for example, we would like to understand also the distribution of the species that we have in the park. So if someone just came as a tourist and go in different area of the park and see something that he don't know what is this, he can just record it. And if we have that information in our database, we can just play and compare what he brings and what we have in our database. And that we can also, we can increase our database in terms of the distribution of the species in the park. And if it's something new, it's good for us also because we can go in that particular place that that person record that information, and that will increase our knowledge about biodiversity in the park.
Right. So to understand the biodiversity in the park, you have all these tools. You can obviously you can collect physical specimens, and you know where you found that organism. You can collect DNA, and but you're also saying that you can add to the database the sounds associated with these species. So you're getting a complete picture of what's going on in the park.
Exactly. We also collect other information like the biology of the species, the habitat preference of that particular species. For example, now we are able, by doing that, we are able, if, for example, if you bring any species for us, and we can able to tell you that this species occur in that particular habitat because we know the biology of that particular species. That's how I came in the Gorongosa National Park in 2013 and participated in the first biodiversity survey.
So you were part of the very first survey. That's really excellent. And I'll just mention now, we're really pleased. You're our inaugural Half Earth Project scholar. And so you're going to continue your education and sort of get work toward an advanced degree in science. And we're really pleased to be able to help support that. That's really great.
Yes, I'm very happy. I'm very happy of that, and I hope that I can continue contributing for documentation of biodiversity.
This katydid was startled, so it displays its wings. It shows, yes. So it shows these patterns here.
Yes, exactly. It's wing just to tell that I'm dangerous. Don't come to me. Yeah. And
so the really interesting thing here, it's as we can see in the background, we have aphids. So normally, as what I was explained, that some species can have two behaviors. So this is the one. So in this case, what will happen here is that because aphid produce sweet sweet substance, so it's called honeydew. So what they do, they just squeeze a little bit, and then on the aphids, and then the aphids produce this honeydew, and they use to eat. But some also use to do trophallaxis. It's meaning they take this honeydew to their halate, so can be a female queen or king. So they need that to develop their wings. So in this particular case, so it's meaning that this species will protect these aphids from the predators. But in the opposite direction, we can also find the same species protecting the plant from other predators. So this is one species that I was talking about.
So they're protecting the aphids. Okay. Now, how about this? This is a very charismatic looking. Is this what do you know what this is?
This is a candidate. This is a nymph of candidates.
I would not have guessed that. Okay.
That it belongs to the genus Ericoryphum. This candidate in particular pretend to be ants. Yeah, this is some kind of,
Okay, it fooled me. I was going to ask Norina, what is this? What kind of ant is this? So that's amazing.
So here we have predation. So we know this is a genus Pachycondyla, so it's a matabili ant. It's really famous ant. So first, because of their behavior. So just to you see, one of the behavior that was discovered that is similar to human is the care. So it's proved now that ants also have care by their sister. If one ant is hurt from the fight, so others will just stop doing what they're doing, and they will take the sister to their colony. And the colony, they give like some chemicals that help that the sister be okay or has a hospital. So what we are seeing here, it's predation between Pachycondyla with termites. So we know that ants use pheromones to communicate. Because of that, people say that ants always know the way back to their colony. So they just use these pheromones. They go, they fight. If something happens, so they will just stop and fight, and then they will take the colony and get back, and then they will continue that. So what happened in the beginning, we were just going out one worker. So we know that workers are infertile females. So one worker will go, and then if she finds the colony of termites, because she just left the trail, she will back to the colonies, tell her sisters, and then they will move like in a 100 of ants going to fight.
So they march.
They return with all the termites.
Okay, so these are the matabele ants you were saying there. That we call them. They're called sometimes army ants. Is that right?
No.
These are different.
Army ants, first, they belong to different subfamilies.
Okay. I'm glad I asked.
And here in Mozambique, if I'm not wrong, we don't have army ants. So army ants, they belong to a different subfamily that it's related to Dorillos. So it's driver ants. So it's related, but they're not same species.
Right.
So because army ants are Aceton busheli and driver ants, we can find another species. We can find Dorillos, Elvolus, and other species. So they are different.
Okay, good. Yeah, but so they're doing these raids. These ants raid termites, termite mounds.
There's a Department of Natural Resources that where we have an agricultural program. For example, we have a coffee program that help the community by getting to get incomes. As we know, we cannot what we are trying to protect the biodiversity, but we would like also to include the community to help us to do that. So that's why we engage the community, but we cannot just say them, don't cut the tree, don't protect the biodiversity.
Right, don't do this, don't do that.
Yeah, yeah. We have to give them some opportunity. That's why we have coffee programs that they can produce a coffee. As we know, we are producing a shade coffee that they can, while they are planting the coffee, they can put also the tree. They can plant also the tree, and in the same time, the tree can grow, and they can have a coffee and sell to the park, and they can have incomes. And we also in the agriculture, we also have other programs, like for example cashew programs, and we have improved honey production here in the scientific department. Each year we have interns that come from the buffer zones, people that finish the high school and they want to go to university, and they come here and work in different sectors like botanical, entomology, molecular biology, and in meantime we invite teachers here that can give them that can teach them to have a that can teach them to be able to be approved at the university.
Thank you so much. I hope to stay in touch and hear more good news as things progress. Thank you very much.
It was a pleasure.
Good.